Recording in progress.
Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera bagi kita semuanya. Dan selamat bergabung dalam acara Guest Lecture Departemen Patologi Klinik FK KMK UGM dengan topik Hemoglobinopathies Decoded, Advancing Molecular Diagnostics and Standardized Screening for Thalassemia and Beyond, serta The Leiden Experience, Advancing Precision in Carrier Detection for Hemoglobinopathies. Bersama saya Primadi Haris Syaifullah, kegiatan guest lecture hari ini akan berlangsung secara luring melalui channel YouTube Patologi Klinik dan Kedokteran Laboratorium UGM. Dan terdapat dua topik menarik yang akan kita nikmati selama kurang lebih 1 jam ke depan. Kami sampaikan kepada para peserta untuk mengisi presensi pada link yang akan kami tampilkan di kolom komentar. Atau bisa juga melalui scan kode kris yang kami tampilkan di layar webinar. Di akhir acara juga akan ada sesi diskusi dan apabila ada pertanyaan bisa disampaikan di kolom komentar. Kegiatan guest lecture ini terselenggara berkat kerjasama seluruh pihak. Pada kesempatan ini, kami selaku panitia mengucapkan sponsor utama, yaitu PT Diagnostics Biotekindo. Dan tak lupa juga kami menyebutkan pihak yang mendukung jalannya acara ini, yaitu Departemen Patologi Klinik FK KMK UGM, Grup Riset Thalassemia, Hibah Riset dan Inovasi untuk Indonesia Maju atau RIIM. Adapun susunan acara pembukaan guest lecture pagi hari ini terdiri atas sambutan oleh Ketua Grup Riset Thalassemia dan dilanjutkan dengan sambutan Ketua Program Studi Patologi Klinik FK KMK UGM sekaligus untuk membuka acara. Guna mengefektifkan waktu, langsung saja kita mulai di acara yang pertama, yaitu sambutan dari Ketua Grup Riset Thalassemia sekaligus mewakili panitia webinar. Kepada Profesor Dr. Niken Satuti Nurhandayani, MSc, kami persilahkan.
Terima kasih. Assalamualaikum warahmatullahi wabarakatuh. Yang terhormat Direktur Pencegahan dan Pengendalian Penyakit Tidak Menular Kementerian Kesehatan RI, Ketua Perhimpunan Dokter Spesialis Patologi Klinik dan Kedokteran Laboratorium Indonesia Cabang Yogyakarta, Ketua Departemen Patologi Klinik dan Kedokteran Laboratorium FKKMK UGM, Ketua Program Studi Patologi Klinik FKKMK UGM, Pembina Grup Riset Thalassemia UGM, para narasumber, moderator, peserta webinar, serta seluruh pihak yang telah berkontribusi dalam terselenggaranya acara ini. Puji dan syukur kehadirat Tuhan Yang Maha Kuasa, karena atas rahmat dan karunia-Nya kita semua dapat berkumpul secara virtual dalam forum ilmiah yang akan dapat membuka ruang dialog lintas disiplin mengenai thalassemia. Suatu webinar yang dirancang sebagai sebuah odyssey, sebuah perjalanan menyeluruh dan reflektif yang tidak hanya membahas aspek diagnostik dan tantangan klinis dari talasemia, tetapi juga menyoroti pendekatan perawatan yang holistik. Karena kita semua menyadari bahwa menghadapi talasemia tidak cukup hanya dengan pendekatan medis, tetapi juga perlu mempertimbangkan aspek psikososial, nutrisi, edukasi keluarga, serta integrasi layanan lintas sektor. It is a great pleasure to welcome Prof. Harteveld from Leiden University Medical Center as our invited speaker. We are excited to gain valuable knowledge from your expertise and experience, and we look forward to the insight and inspiration you will share with us today. Apresiasi kepada seluruh pembicara, panitia, sponsor, dan para peserta yang telah meluangkan waktu untuk webinar ini. Semoga diskusi hari ini membawa manfaat yang luas dan menjadi pemantik kolaborasi lebih lanjut untuk masa depan yang lebih baik bagi para penyintas thalassemia. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh.
Kami ucapkan terima kasih kepada Profesor Niken atas sambutannya. Berikutnya adalah sambutan dari Ketua Program Studi Patologi Klinik FK KMK UGM, sekaligus membuka acara guest lecture pagi hari ini. Kepada Dr. Teguh Triyono, MKes SPPK Konsultan, kami persilakan.
Terima kasih, selamat pagi. Assalamualaikum warahmatullahi wabarakatuh. Yang kami hormati Ibu Direktur Pencegahan dan Pengendalian Penyakit Tidak Menular Kementerian Kesehatan Republik Indonesia, Ketua atau Pembina Thalassemia Research Group, Ketua Cabang PDS Patkin Yogyakarta, Ketua Departemen Patologi Klinik Fakultas Kedokteran dan Kesehatan Masyarakat FK KMK UGM yang kami hormati, sekaligus Ketua Panitia rasanya ini. Juga Bapak Ibu sekalian, peserta, seluruh peserta kegiatan guest lecture pada pagi hari ini. Pertama-tama tentu kita tidak pernah lupa bahwa untuk selalu memanjatkan puji syukur kehadirat Allah SWT, kita senantiasa diberikan nikmat, salah satunya nikmat kesehatan dan nikmat waktu, sehingga pagi hari ini kita bisa bersama untuk menyertai, mengikuti kegiatan guest lecture pada pagi hari ini. Kegiatan ini merupakan satu kegiatan yang menurut saya multi aspek. Thalassemia merupakan penyakit yang melibatkan multidisiplin dan tidak hanya medis saja, psikologi, sosial, dan yang lainnya. Jadi semuanya akan terlibat. Sehingga kalau kita lihat topik pagi hari ini, Comprehensive Odyssey Through Thalassemia, Diagnostic Challenges and also Holistic Care, sesuatu yang sangat tepat dan sangat penting bagi kita semuanya. Terima kasih kepada para seluruh pembicara, juga tentu saja moderator, dan sekali lagi juga selamat kepada seluruh panitia atas kesuksesan kerja pada pagi hari ini. Topik ini topik yang sangat penting sekali lagi, dan also it is a honor for us to have our distinguished speaker, Prof. Harteveld here. Thank you so much for here and for giving us your experiences and your insight for us. Thank you so much, and also thank you Mr. Greg here also to give your experiences for us in Indonesia. Saya kira itu yang bisa saya sampaikan. Sekali lagi terima kasih kepada semuanya, sehingga acara pagi ini bisa berjalan dengan sangat baik, dan tentu saja harapannya bisa menambah wawasan kita semuanya untuk peningkatan pelayanan kesehatan di Indonesia, khususnya di bidang thalassemia. Itu saja dari saya, terima kasih. Assalamualaikum warahmatullahi wabarakatuh. Ternyata harus membuka. Baik, dengan bismillahirrahmanirrahim, acara kegiatan guest lecture pada pagi hari ini kita nyatakan dibuka. Terima kasih.
Kami ucapkan terima kasih kepada Dr. Teguh atas sambutannya. Tidak lupa kami informasikan kepada para peserta bahwa di akhir acara akan diadakan foto bersama seluruh peserta guest lecture secara daring. Ladies and gentlemen, we proceed to the first session that will be presented with topic Hemoglobinopathies Decoded, Advancing Molecular Diagnostics and Standardized Screening for Thalassemia. And continued by the second topic, the Leiden Experience Advancing Precision in Carrier Detection for Hemoglobinopathies, that will be delivered by Associate Professor Dr. Cornelis R. Houteveld. He is an associate professor and senior researcher in clinical genetics and hemoglobinopathy. This session also will be guided by chairperson Dr. Nur Ima Fatimah Harahap, PhD SPPK. Her curriculum vitae is shown on your screen right now. For Dr. Ima, time is yours.
Thank you very much, Master of Ceremony. Good morning. On behalf of the Department of Laboratory Medicine and Clinical Laboratory and also Thalassemia Research Group Universitas Gadjah Mada, I warmly welcome you to join our guest lecture titled A Comprehensive Odyssey Through Thalassemia, Diagnostics, Challenges, and Holistic Care. And for this morning's session, we will have a very impressive lectures from expert in Netherlands. Actually, this event is held as commemoration of Thalassemia International Day, which was celebrated on the May 8th, and also as part of our commitment of thalassemia in Indonesia. But before that, I would like to show you the curriculum vitae of our speaker for this morning session. Actually, our speaker is an impressive expert from the Netherlands, and you can show his curriculum vitae on the display. He is currently working as a part of staff member in Department of Clinical Genetics in Leiden University Medical Center. As already shown in his curriculum vitae, our speaker today, Dr. Cornelis Leonard Houteveld, PhD Associate Professor, he already shown impressive and also extensive work in the diagnostic of thalassemia internationally, and also his work have been cited for researchers and also thalassemia care in Indonesia. And without further ado, I will also welcome to Dr. Leonard Houteveld to the floor. Yeah, thank you very much. But maybe before that, I would like to remind our participant, if you have any question during his presentation, you can give us your question by sending your name, your full name, and also your question in brief and compact format. So the floor is yours, Dr. Houteveld. Thank you.
Thank you, Mrs. Chair, and I thank you for your very kind words. I would like to thank the organizers for inviting me to give me the opportunity to give a talk about the work I've done in Leiden together with my group. And I know it sounds a bit strange that someone from a country where thalassemia is not endemic at all is going to give a talk about thalassemia. But still, I would like to share and take you along a journey which might show you sometimes unexpected mechanisms. Yes, thank you. So on the first slide, you will see my research building, and next to it are the tulip fields, which are the famous Keukenhof. For those who have visited the Netherlands, this is a botanical garden where all the tulips blossom in April and May. I have nothing to disclose. The thalassemias are a multisystemic disease, and these are diseases of the hemoglobin. Hemoglobin are tetrameric proteins responsible for the oxygen transport in the body. The hemoglobin A, adult hemoglobin A, consists of alpha-2, beta-2. In thalassemia, especially beta thalassemia major, there is a reduced synthesis of the beta chain, and this means that there is an excess of alpha synthesis, and this forms, these alpha globins form tetramers with itself, and that precipitates, destroys the membrane, and causes ineffective erythropoiesis and severe hemolytic anemia. Sickle cell disease, which is much more frequent in the Netherlands, is a totally different disease. The mutation is actually a single mutation, changing a glutamic acid position 6 of the beta chain to a valine, and this gives the hemoglobin the habit to form polymers when the oxygen is there's oxygen depletion of the hemoglobin S, and this distorts the red cell to the typical sickle shape, and this causes infarction of the small vesicles and vaso-occlusive crisis. Hemoglobinopathies are very prevalent in areas where also malaria is endemic, and malaria is spread by the Anopheles because the Anopheles carries also the parasite Plasmodium falciparum, and that is the cause of malaria tropica, the severe form of malaria, which causes the death of many young children. However, carriers have a protection against early death by malaria. They have a higher chance of surviving. And this means that we see in the map regions where typical hemoglobinopathies occur. So like hemoglobin C, which is more prevalent in West Africa, S, which has different geographical locations, thalassemia, which is the yellow band. Of course, there is region specificity in the mutations which occur, and hemoglobin E, which is very frequent in Southeast Asia. Thalassemia is caused by pathogenic variants in the HbA1, HbA2, and HbB genes. As is indicated, they are located in gene families, which are on the short arm of chromosome 16, where the alpha globin gene cluster resides, and on chromosome 11, where we can see the beta globin gene cluster. Also is depicted in this figure the major conserved sequence. This is a regulatory element which is responsible for the switch of the zeta and epsilon and alpha-1 expression during a tissue-specific and developmental specific way. Similar, we see at the beta globin gene cluster, the beta LCR, which is let's say doing the switch from epsilon to G gamma expression in the embryonic to fetal phase, up to the delta and beta expression in the adult phase. So as adults, we have to deal with the expression of the alpha genes and the beta gene, and both alpha genes are equally highly expressed as the single beta gene. So there is a balance between alpha synthesis and beta synthesis. And there is a slight expression of the delta gene, and that's because the delta has a very poor promoter. The majority of our hemoglobin in adults is hemoglobin A, 97.5%, and a minority is hemoglobin A2, 2.5%, but that is a very interesting marker to detect beta thalassemia carriers. The switch graphic shows when the genes are expressed and at what level during the different stages in development. So in the embryonic phase, hemoglobin GAWA1 is formed because the zeta and the epsilon genes are active. And throughout the development, during the fetal period, there is mainly expression of hemoglobin F, or synthesis of hemoglobin F, due to this expression of alpha and the gamma genes. And around birth, there's a steep decrease of gamma synthesis, because the gamma genes are less expressed and the expression is taken over by the beta genes. So in this case, you can imagine that if there's a problem with the beta gene, like sickle cell disease or beta thalassemia major, this becomes evident after 6 months after birth, when the expression is fully relying on the expression of the beta gene. Well, the alpha thalassemias, they might become problematic much earlier in development because of the deficiency or the absence of alpha gene expression. And in that case, the last hemoglobin which can be made is hemoglobin Portland, and that is zeta to gamma 2. And this is responsible for, let's say, the survival of the fetus up to the end of the gestation, and then it will unfortunately die of severe hemolysis, severe hemolytic anemia, or shortly after birth. So this is the hemoglobin Barr's hydrops fetalis syndrome, which is very frequent in Southeast Asia. So it is very important to detect carriers, because if you detect carriers, you can inform them about the genetic risk of having a baby with a sickle cell disease or beta thalassemia major or alpha thalassemia major. Carriers are healthy and often unaware of the genetic risk of thalassemia or sickle cell disease. So how do we perform the diagnostics for hemoglobinopathies to detect carriers? Well, these are 4 jigsaw puzzle pieces, and they all have to fit. So you start out with the red cell hematology. You subsequently do hemoglobin separation or hemoglobin typing. You do DNA analysis, and this whole picture should match the clinical diagnosis. And if not, then you might have missed something. You have to repeat the cycle. This jigsaw puzzle can also be presented in a flowchart. Flowcharts are, let's say, effective for laboratory personnel. They follow the strict regulations, and they can divide, let's say, the hematology from the HPLC and DNA analysis with the certain cutoff values which are indicated here. However, this could be also presented in more detail, let's say. So this is the hematology. You look at anemia, microcytic hyperchromia, and to the red blood cell counts. Now, if we look more closely to the red cell hematology, then we see something remarkable if we go round with the clock. So if we start left upper side with the graph of hemoglobin, and you concentrate on the middle column, then you see that there is a distribution range of hemoglobin, which is in the carrier quite similar to the normal control, which is on the left. The patient, of course, has a very, very low hemoglobin, very typical for thalassemia major. So if we concentrate on the middle one, you see that the carrier shows overlap with the normal hemoglobin. If we go to the right, you see the MCV. The MCV is the mean cellular volume, and you see that there is a clear distinction. So the MCV is lower in the carrier in comparison to the normal. If we go down, then we see the MCH, which is the mean hemoglobin content in the cell. And you see in the middle one that there is little overlap with the normal. So the red cells, which we are looking at, are smaller than usual and more deprived of hemoglobin than usual. Now we move to the left lower picture, the red blood cell count, and now you see something remarkable. If you concentrate on the carrier in the middle, you see that there's an elevation of red blood cell count. So in spite of the fact that the red cells are smaller than usual and more deprived of hemoglobin, there are more of them. So there's a compensatory effect, and that explains, in fact, why the hemoglobin in the upper left corner is actually almost normal for the carrier. This also explains why being a carrier is so fortunate. You are protected against death by malaria, and still you have enough hemoglobin to do all the work you need to do. Hemoglobin separation is very important in the hemoglobinopathies diagnostics because it can identify very easily hemoglobin variants, like hemoglobin S, for instance, and that can be done on HPLC, which is the upper part, or capillary electrophoresis, which is the lower part of the figure. And both systems can also detect the elevated hemoglobin A2, 5.6%, doubled from the 2.5, which is very typical for the beta thalassemia carrier. The third part is DNA analysis, and usually we do it sequential. So we look at the gap PCR for the common alpha thalassemia deletions. This solves up to 80% of all the deletions present, because the majority of the world population carries 3.7, one typical mutation, or Southeast Asian 4.2, and a number of others. Then the second possibility is that you have a point mutation. In that case, this accounts for 20% or 90% of the non-deletion variants in alpha thalassemia, and less than 1% is uncommon deletions, which can be identified with the copy number variation using MLPA. Beta thalassemia, on the other hand, the majority are point mutations, so non-deletion variants in 95%. So you start out with Sanger sequencing in that case, and the subsequent 5% are uncommon CNVs seen by MLPA. So if we make it more simple, you have the clinical picture, the patient in the middle, you do red cell hematology, HPLC typing, and DNA analysis. The upper part is in fact your phenotype, and the lower part is the genotype. And the genotype should explain the phenotype, or there should be phenotype-genotype correlation. Now I'll show you 3 examples of let's say, a common diagnosis for case 1. So there's a lady, 42 years old. She's of African-American descendants, and we see that she is having a normal hematology, because abnormalities are indicated in red. So she's normochromic, normocytic, and she has oh, that's interesting. She doesn't have a normal separation. She has an extra band on the S position. So apparently she is carrier of hemoglobin S. And the amount of S is 40%. That is already indicative that she is having a S trait. And indeed, DNA sequencing confirms this. And she is negative for the common alpha thalassemia deletions. So this case has been solved quite easily. Now the second case, we see a lady of 29 years old. She was the mother of a child who appeared positive in the HEAL-PIC screening, and she shows a microcytic hyperchromia and an abnormal pattern with 34% hemoglobin S. If we do DNA analysis for this one, we might expect an alpha thalassemia, because alpha thalassemia has an influence on the S percentage. Let's look at the S. This is a normal S carrier. Here you can see the number of alpha genes, and there's one beta S gene indicated in yellow. So the hemoglobin S migrates between the A and the A2 on capillary electrophoresis, and the amount is 40%. However, when you co-inherit an alpha plus thalassemia, the S percentage is decreasing up to 34%. And if you miss 2 alpha genes, either in cis or in trans, the S drops even to 25%. You can almost draw a line. So in this case, with 34%, it's most likely that she has an additional alpha thalassemia defect. And indeed, this was demonstrated by the gap PCR. Right. Well, in order of time, I'll skip this one, because this is a very regular S carrier, which you can see from the elevated A2. But what does happen if the phenotype doesn't fit the genotype? Well, then it starts to become interesting. And I have 3 examples which I have labeled everything depends on balance, late onset thalassemia, transfusion-dependent thalassemia intermedia, and it looks like a horse, it smells like a horse, and yet. Well, let's proceed very quickly because time is running out. We have a patient which is 30 years old, and she is diagnosed with a thalassemia intermedia at the age of 3. The second family, second lady, was also even transfusion dependent. So both appear to be carriers of a beta thalassemia, which is in contradiction with the severity of their clinic. So why is that? We cannot explain it simply by the presence of an alpha triplication and the presence of a single beta mutation. A carrier should have been asymptomatic. Well, at the time that we investigated these patients, we just published the MLPA technique in 2005. And in this graph, it shows that you can detect differences in ratio when a deletion is present, very easily by simply looking at the different colors and the ratios in comparison with a normal control. When we published this, our Italian colleagues wanted to have these 2 cases investigated, and they sent it to us together with a student. And when we investigated this, we found a remarkable elevation of the ratio, which is the contradiction of a deletion. So there was a duplication of the entire alpha globin gene cluster. And if you combine that with a deficiency of a single beta, then you get an unbalance which is much larger than it used to be. If you have a normal situation, you have equal amount of alpha and beta. If you have a beta thalassemia trait, the amount of alpha is a bit too high, but it can still be reduced by proteolytic processes in the cells. However, if the alpha triplication is, or there's an alpha multiplication, the balance becomes too high and the patient becomes transfusion dependent. We have published this with the question, are there more common modifiers? And yes, after our publication, there were many more, which appeared to have a segmental duplication, which is now more easy to detect using MLPA. Right, then I go to the late onset transfusion-dependent thalassemia. These patients encircled, they have a late onset of beta thalassemia. They were born as carriers, but they developed a transfusion-dependent beta thalassemia major later in life. And this is due to another phenomenon. So we have to see they became anemic, well, they were born as carriers, but they became severely anemic in the 3rd and 4th decade of life. And if we look carefully at their sequence patterns, then we see that there is a slight presence of the normal allele. So these patients look mosaic. And where does this mosaicism come from? Well, we did a SNP analysis using the 730,000 SNPs all over the genome using the Infinium assay. And then you can detect either homozygosity, heterozygosity, or of the different SNPs. Well, if we look at the normal distribution of SNPs along chromosome 11, this is a normal picture. This is what you expect. However, if we look at the patient, Propositus 1, we see that this patient suffers from an uniparental isodisomy of the short arm of chromosome 11, which contains the beta gene inherited from the father. And if you remember, the father was also the one who carried the beta tau. All the other 2 patients, so all 3 of them, had an uniparental disomy of a different percentage, and they expressed all the beta thalassemia major later in life. But how come that this clone which carries this UPD is selected? Well, that is probably due to a growth advantage, which is given by the presence of IGF-2 H19 in linkage with the beta gene. In other words, this is something we have seen in Beckwith-Wiedemann syndrome, a fetal overgrowth, which is an imprinting-sensitive story. So the paternal allele expresses IGF-2 and the maternal allele expresses H19. The paternal allele is the one which carries the mutation. In heterozygosity, there is a balance. However, in our patients, there's UPD of the paternal allele. So there's overexpression of IGF-2. Well, this system would also work in the opposite direction. That is the loss of the maternal allele. And this graph shows a presentation of deletions of the maternal allele, which also gives a late onset of beta thalassemia major. The last sample is 2 Dutch families which have beta thalassemia trait, which is very obvious from the fact that they have an elevated A2 and a microcytic hyperchromic picture. However, there is no mutation in the beta gene, and that's quite remarkable. Why is there something? Are we missing something? Well, yes, we did. And therefore we needed whole exome sequencing because whole exome sequencing showed that these patients in the family 1, they had one particular variant in common. And that is the erythropoietin specific expression parameter, the protein SUB5H or the gene SUB5H, which codes for SPUT5. And all the carriers of the trait, they had actually this mutation in the splice donor site, which was disrupted by this mutation. The second Dutch family had a different mutation, but in the same gene. So RNA analysis showed that this gave a premature stop. And the combination in 2 Greek families of a SUB5H mutation together with a beta mutation explains in these patients the more severe phenotype, which we have seen in the patients circled in green. Up to date, there are now more than 30 different mutations found in the SUB5H. At the time, we only had 8, but now worldwide, 30 different mutations have been found. And the question remains, why is this happening? What is this protein? Well, it is part of the DCIF complex. SPUT5 and SPUT4, they form a dimer, which is part of the RNA polymerase II complex, regulating elongation and blocking of elongation, so of transcription. So this is a very important protein, but those people who are haploinsufficient for this, they also have a decreased beta synthesis. And this is probably because there is an influence on GATA1 induction. In conclusion, I can say carrier detection for hemoglobinopathies is the key element in the prevention of thalassemia of major and sickle cell disease. A diagnostic flowchart in which you combine red cell hematology, hemoglobin separation and DNA analysis reveals the majority of the molecular causes, but a deviation of the standard holds the key for unraveling uncommon molecular mechanisms. And often novel findings are technology driven. Sometimes you have to wait for decades before the right technology pops up so that you can, let's say, unravel this mystery. So to discover new things requires focus, a good team, international collaboration, of course the collaboration of patients, the family of patients and friends in science. I'd like to acknowledge my group and my foreign collaborators who have, through which friendship and collaboration, we were able to do all this kind of beautiful things. Thank you very much. Okay, yes.
Thank you very much, Dr. Harteveld. So maybe you can continue to the second lecture.
Yes, of course.
Directly before before we move forward to discussion session.
Okay, the second lecture is the Leiden experience. So I think the major thing I would like to discuss here is what is the prevalence of hemoglobinopathy in the Netherlands? How do we deal with the carrier detection? Well, as said in the previous talk, we have beta thalassemias and alpha thalassemias and sickle cell disease, but sickle cell disease is more common in the Netherlands. And that is a totally different disease from thalassemia, because thalassemia is more, let's say, chronic anemia, and sickle cell disease is characterized by periods of pain, extreme pain during a crisis. But in between, things go relatively all right up to the second or of the following crisis. So that is a different proceeding of the disease, but both are multisystemic diseases which are very severe. The prevalence of hemoglobinopathy in the Netherlands is, of course, very low in comparison to Southeast Asia and in comparison to Indonesia. However, if we look at the geography, 18 million inhabitants in the Netherlands, and Netherlands are quite small. But because of our history, we have an increasing number of thalassemias in our population. And that starts with the colonization history of the Netherlands. The Netherlands had colonies in South America, especially Suriname and the Antilles. And during the times of slavery, slaves were brought from West Africa to South America to work on the plants. When slavery was abolished, then contract workers were attracted from India at very low wages. The situation was very, very bad. But people immigrated to these colonies, and they intermixed. So you had intermixture of people from India, from Africa, from Spain, from China, and from South Africa. And this mixture also contained not only hemoglobin S, but also many variants of alpha and beta thalassemia. And in the 1970s, there was a lot of migration from Suriname to the Netherlands because of the independence of Suriname. Many people from Suriname still had a Dutch passport, and they went to the Netherlands for economical reasons. We also had colonies in South Africa, and as you are very well aware of, the Dutch were also in Indonesia. And this contact with Indonesia, there are also a lot of people who immigrated from Indonesia to the Netherlands, and that bringing hemoglobin E and beta thalassemia and alpha thalassemia. And also recently, people from Africa and from let's say other European countries, because the Netherlands were also overrun by our neighbors, the Germans, the French, the Spanish. The Netherlands have been for a very long time be part of the Hispanic Empire, and in the 1600s we had our liberation war against the Spanish, and in that period a lot of soldiers came to the Netherlands, Spanish soldiers, Italian soldiers, and they all left a little bit of their genes. So you can imagine that also people who are Dutch or think they are Dutch may have an ancestor coming from Indonesia or from Spain or from the Mediterranean area. And sometimes they are not aware of it. So even if there is a mixed marriage, we say, please do check the partner, because if you have one partner who is a carrier of S or beta thalassemia and is married to a Dutchman or a Dutch woman, don't think that they cannot have thalassemia or sickle cell. And very recently, we have many immigrants from Syria and also from Turkey and Morocco. So it is a very mixed population. So if we look at the statistics, we see that 1.9 million people have an immigrant background of the 18 million inhabitants. 43% are of Western origin, and usually they don't have the hemoglobinopathy genes. However, the rest is from areas where there is a higher endemicity for hemoglobinopathies. And this results in 35 to 40 neonates per year with sickle cell disease, and 8 to 10 about neonates per year with beta thalassemia intermedia or major, and 1 or 2 with hemoglobin H disease. So approximately we have 1,500 patients, and we don't know exactly because there is no national registration for patients. And we have approximately more than 100,000 carriers, and most of them are unaware of being a carrier. So approximately 1 in 10 people with an immigrant population from an hemoglobinopathy endemic area is a carrier. So that is relatively relatively high. It's not as high as in Indonesia for sure, but it is something to take into account. So carriers are healthy, And when do you do carrier detection? Well, that can be preconceptional. That will be the best moment. Then you can think about the risk for having babies with sickle cell disease, and you can do maybe pre-implantation diagnostics or abortion on medical indication. You can also offer carrier detection in the first intake in pregnancy, as is done in the UK, antenatal screening programs, but we don't have that in the Netherlands. Only midwives do it by their own initiative. And neonatal screening, this is the only thing which is organized in the Netherlands. We have a screening program for babies with sickle cell disease and beta thalassemia major. So for the diagnosis, I will not go into that again because we have seen that in the previous talk, but we look at hematology and HPLC with the different equipment, and this is the moment that we can see abnormalities like beta thalassemia traits or carriership of an hemoglobin variant. This is an example. Here you can see a more, let's say, difficult pattern in which the A is a little bit low and there is apparently an S peak which is quite high. So this looks a little bit like a combination of a S carrier who also has a beta plus thalassemia on top of it. Well, let's have a look at the hematology. Indeed, we see a low hemoglobin, low red blood cell count, and low PCV, which means hemolytic anemia. So this lady has a hemolytic anemia. And if you detect the S, you know that she is also an S carrier. So from this, we can do the DNA analysis and compare the results. So indeed, when we did gap PCR, we saw she was carrier of a 3.7 deletion. When sequencing the beta, we found two defects. The S was confirmed, and we also saw a beta plus thalassemia mutation, which is in the promoter region of the beta gene. Well, the alpha 3.7 can easily occur by mispairing and homologous recombination between the homology boxes in which the alpha genes are embedded. So during meiosis 1, you can have a crossover in mismatched strands of the chromosomes. And this results in the loss of an alpha gene and a gain on the other possibility, alpha triplication, but the alpha 3.7 is selected in malarial areas. If we draw the picture of the genotype of this lady, then we see that she has 3 alpha genes. And if we look at the beta globin gene cluster, we notice that she expresses F and also S. So, in fact, because of the mutations present, we see that she must have a mutation in the beta gene, but now it blocks. I don't know why. Oh, yeah, there it is. So we can see that she has one mutated beta gene, which is expressed at a lower level, and she has an S allele, which is expressed at a normal level. So how many tetramers could you make from this? Well, the alpha is expressed but less, and then you have the beta. So we see hemoglobin A and hemoglobin A2. But besides that, we also have alpha 2 beta S2, which is hemoglobin S, and alpha 2 gamma 2, which is hemoglobin F. And all these percentages match perfectly with the picture on the left. So this is what we can expect. Right. And from the DNA sequencing, we now go to the next generation sequencing. Next generation sequencing can be applied, for instance, by looking at the whole exome. Well, the whole exome and whole genome and targeted are frequently used in diagnostics. So here we can see the representation of the whole genome sequencing, which seems, well, then you take, you sequence the complete human genome. You can indicate that this as the whole apple. But you can also do whole exome sequencing. Then you only looked at the expressed parts of it. So that's only 1% of the total. You could imagine that as a slice of the apple. But another approach would be targeted sequencing, and then you look just at the genes of interest. So this could be imagined by little pieces of the slice of the apple. And the different approach depends on what is the question. So for instance, if you have rare anemia disorders, you could also decide to use a gene panel to screen for common genes involved in the hematopoiesis. And this was done for 5 cases with an unexplained severe chronic anemia, some without family history of this trait. So we have a dominant trait, which was not expected because the parents were unaffected, a targeted NGS by using the disease panel for red anemia disorders was done for cases 1, 2, and 3, which I'm going to show you in a second. And the trio analysis using whole exome sequencing was performed in case 4 and 5. So let's first go to case 1, 2, and 3. Well, here there was a remarkable low hemoglobin and MCV and MCH, and it was only found in this patient, and there was not a clear indication from the family, except for maybe case 3, in which the mother was diagnosed with hereditary spherocytosis. When using the targeted approach, we had a gene panel with a lot of genes represented, and doing these samples, it appeared that case 1 was a hemoglobin Bristol alesia, which is a de novo mutation in this patient, and which is highly unstable, explaining the red cell anemia, hemolytic anemia. The second case was a hemoglobin Evans, and that is an alpha variant. This one should be inherited, but the parents were not analyzed. That's probably the reason why it was not picked up. And the third case was in hemoglobin de Bruce. Also, that one is highly unstable, giving rise to erythrocytosis, and was not suspected in the mother. Case 4 and 5 were investigated doing whole exome sequencing. This is usually done in a trio example. So there are 2 possibilities in this case. Either you have 2 normal parents and the child carries a dominant mutation which occurred de novo, or the parents are both carriers of an autosomal recessive trait and which comes together in one of the children. And of course that has consequences for a next pregnancy. When we did trio analysis for the 2 cases indicated, it appeared that this was the case. Both were de novo, and they were de novo mutations in the beta globin gene, in which the stop codon was changed to a coding codon, which caused an elongated beta globin chain, which is giving rise to an unstable hemoglobin. The one was the hemoglobin Zunyi, which was previously published, and the other was hemoglobin Mokum. It was in the Dutch family, and that was not published yet. So we have presented this mutation also to the ETA gene database. Both result in an extension, even though the amino acid order is a little bit different. Example 2 is a complex breakpoint, which we have investigated by whole genome sequencing. It was a family in which the father was carrier of a beta plus thalassemia mutation, which he gave to his children, but his children were much more severely affected. They had an intermediate phenotype, which was unexpected. So we did MLPA analysis of the alpha globin gene cluster, and we saw in these 2 children that there was an increased ratio of the alpha globin gene cluster, which means that there are multiple alpha genes present. If we look at the mother who may have given this to the children, it is remarkable that she doesn't show this ratio increase. And that is because she is mosaic. She is mosaic for the multiplication of the alpha, probably in the germline. And that's why she transmitted it to 3 of her children. The segmental triplication of the entire gene cluster aggravates the alpha-beta ratio imbalance in the beta thalassemia carriers. And we have now seen a couple of examples of the segmental duplication of the alpha globin genes, but this one is an alpha triplication. That is the first one that we see as we believe. So there are different breakpoints for those which should be analyzed, and this can be done with whole genome sequencing. Here you see the Nexus clinical data, which you can very markedly see the position of the 2 different breakpoints. And more in detail, the sequence analysis reveals that you see a kind of rainbow sequences in the 4 colors, representing the A, the C, the G, and the T. And these fragments, which show this typical rainbow sequence, is the breakpoint fragment. And the breakpoint fragment that is, let's say, crossing the bridge between the normal sequence and the sequence on the other side. So this, the duplicated sequence or the segmental triplicated sequence. And we did this for the first breakpoint, the second breakpoint, and the third breakpoint, which gives in total the resolution of a very complex map of the segmental triplication, because there were also some orphan sequences within the breakpoint regions. Well, if you want to do next generation sequencing for a routine application, you need the standardized protocol and standardized kits. And especially commercial kits are important in this, because you hope that they will have the IVDR qualification, which is important for the quality assessment. Well, Devyser offers a solution for the alpha and beta globin genes. They make use of a targeted approach in which the amplicons are labeled in a specific way so that you can distinguish your different patients, so you can work in libraries and in pools. And they cover not only the alpha-2 and alpha-1 and the beta gene, but also they have couples of primers in the clusters for alpha and beta, so that you can do copy number variant analysis as well. They also included gap PCR primers for some specific deletions in the alpha cluster and in the beta cluster. And this, I'll show you some examples of the Devyser results from our validation. You see here the beta globin gene cluster on the left, the alpha globin gene cluster on the right, and this is a deletion which coincides with the thalassemia deletion, a tie deletion. The tie deletion is indicated in green, which means that it is a gap PCR result, which proves that this is really tie. But again, you can also see it from the copy number variation. So you see a decreased ratio for the probes allocated in this alpha gene cluster, matching the tie deletion. Similarly, you can also identify the 3.7 deletion, even though there are not primers in this version of the kit to detect the 3.7. Devyser, well, we told Devyser that we were not happy with that, but the Devyser said that they were working on a solution and that this will be included in version 2, which will appear next year, the first quarter or second quarter of next year. So we will be waiting for version 2 then. But from the copy number variation, you can very well appreciate the presence of the homozygosity for the 3.7 and the presence of a alpha triplication. So using this targeted approach, we try to do the routine diagnostics for carrier detection. So now I show you an example in which we have identified a hemoglobin S carrier. That is very obvious from the listing of the SNPs which were found in the beta gene. So the C.28-T is indicative for the hemoglobin S. But what we also see is that there is a mutation in the alpha and that is the C dot minus 59 C to T. However, the system is not able to allocate it on alpha 1 or on alpha 2. And that is because we use this system uses short reads. And short reads in two homologous genes gives problems because then it cannot allocate the one or the other. To distinguish these two is too difficult because it's only short reads. So in this case, you would still need Sanger sequencing to confirm whether on which gene it is located. Well, in this case, it's not so important because this is a polymorphism. So we also had one example of a diagnosis which was missed by the old school sequential approach using Sanger sequencing and gap PCR and MLPA, but was detected using divisor. And this is a patient, a 47 years old female, who presented with this capillary electrophoresis pattern, which indicates a suspected hemoglobin Bart's, an A, F, A2, 2.5, and something in the C zone, which was equivalent to constant spring. So it's a combination of Southeast Asian constant spring, which we identified with our old school sequential approach. However, when we did this on the divisor kit, it revealed that there was an extra mutation in the beta, which we missed by not looking at the beta globin gene because we didn't have any clue to do that. But it still was there, and this is showing that sometimes next generation sequencing is more powerful than your sequential methods. The benefits of next generation sequencing, well, we know the first generation sequencing, the Sanger sequencing. Then you need a sequential approach, gene by gene, dependent on the hematology, HPLC and CE. The second generation makes use of next generation short reads. That means that you can use targeted panels, you can have selected genes, like for HbA1, HbA2, and HbB, like in the divisor kit. You can have whole exome sequencing. This is usually if you have unknown causes of hemolytic anemia, or whole genome sequencing. And that is usually done for characterization of unknown deletions or duplication breakpoints. We are heading now for the third generation sequencing, and that is long reads. Well, then you have no longer the problem of the homologous alpha 1 and alpha 2 gene because you can do them in one go. You can do the phasing. And also you could detect complex rearrangements more easily. The future perspectives are that genetic labs will make more use of NGS, and we will also have more incidental findings. That takes time to solve those variants of uncertain significance. And then geneticists can't be expert in everything. Next generation sequencing driven results require consultancy of experts to correlate the variant and the clinical phenotype, which is not always obvious for a geneticist who is doing, let's say, thousands of different genetic diseases on a day. I would like to thank you, terima kasih banyak, and Dutch. Dank voor uw aandacht.
Thank you very much. Very nice presentation, Dr. Harteveld. Please now have a seat. We'll now enter our discussion session, and I just got information from our committee that we have several questions, and because we have so limited time, we will summarize those questions into some of maybe two or three questions. And to the committee, please kindly show the questions, and I will read and give to Dr. Harteveld to respond to. To the committee, please show the questions. Okay, maybe this is the first question. Oh yes, before that, I already informed that we need identity, the name, and affiliation. But because we summarize the questions, so we only choose the most common question delivered from the audience. And the first question, I will read in English language. As explained before, to discover new things requires focus, a good team, international collaboration, patient families, and friends. What's your opinion or experience from those aspects? Which is the major role for discover new things? And would you explain why?
Yes, well, I think the major role for discovering new things is when you develop the let's say the feature of understanding this is not explaining the whole thing. If you do hematology and hemoglobin typing and DNA analysis and you still have no explanation for the clinical severity, that is the moment that you must think, oh, well, I might be missing something here. And to have more information on that, you need to go back to the doctor. You have to go back to the clinician and ask, what can you describe what is the problem? And can you help me think why the DNA, the genotype which you have found, does not match this pattern? And if that is the case, then of course you have to come up with an hypothesis. Usually the hypothesis is that there's something else in the genome which is responsible for this. And previously we didn't have any possibility to investigate this. However, with the advent of next generation sequencing and the let's say the software becomes much more clever every time, you have finally also the technical capacity to go further and start looking for this particular modifying factor. I think that is very important. Secondly is the family. You need a family pedigree and a large pedigree, preferably 3 generations. If that is possible and you have more affected members, it will be easier to exclude variants of uncertain significance, because everything which let's say the members have in common is a potential candidate, but what they don't have in common can be excluded. So in this way, that is also why the patient comes in and the family of patients comes in. And then friends, networking is very important. To make friends in science is important, because we don't have very interesting cases in the Netherlands, I must admit. Often we only have a single case, but more cases make a mechanism. So you have to look abroad. You have to look with your friends. Ask them, this is what I find. Do you have something similar? And if yes, may I investigate it with our new technology? And in this case, you can find more cases and discover mechanisms.
Okay, very interesting for the answer. I will try to translate in our bahasa Indonesia. Untuk pertanyaan ini sebenarnya ketika terjadi kasus yang sulit, ini memang apabila terdapat kasus yang memang klinisnya tidak sesuai dengan hematologi atau HB typing atau memang analisis genetik, mungkin harus dipertimbangkan kita dari laboratorium berkomunikasi dengan klinisi, apakah tanda dan gejala pasien apakah spesifik ke arah thalassemia atau hemoglobinopathy atau tidak. Dan apabila memang mendukung, memang lebih baik kita melakukan analisis lebih dalam untuk genetiknya. I'm sorry, I have to respond in our bahasa Indonesia. Dan apabila memang terjadi kesulitan, mungkin kita bisa bekerja sama dengan pihak lain yang memang punya pengalaman lebih, dan kita bisa sharing juga dengan klinisi lain atau laboratorium lain yang mungkin kasus ini tidak ketemu di kita tetapi ketemu di tempat lain. Nah, ini juga pentingnya untuk berkolaborasi bersama, karena apabila kita menemukan kasus yang sulit, kita bisa bertanya atau meminta pendapat dari sejawat yang lain. Dan juga setelah tadi kita mengkonfirmasi klinis pasien, kemudian juga bertanya dengan sejawat, tidak lupa juga kita harus bertanya untuk riwayat penyakit di keluarga, apakah ada anggota keluarga yang memang memiliki keluhan serupa, dan ini memang dilihat dari pedigree atau memang riwayat keluarga, dan ini memang dilihat dalam 3 tingkatan, mungkin dari pasien, kemudian orang tua, dan orang tuanya lagi ke atas. Mungkin ini untuk pertanyaan pertama, so we will continue to our second questions. Please, so okay, this is from the question. Second question is from Yona Mimanda. Okay, so hello, Dr. Harteveld. Thank you for your excellent lecture today. It was both insightful and inspiring. In my research, I have encountered cases where beta thalassemia and iron deficiency anemia coexist. Yes, this is actually really common in Indonesia. Some literature suggests that iron deficiency anemia may reduce HbA2 levels. This could complicate the screening process, especially since iron supplementation prior to screening is not always practical. How significant is the impact of iron deficiency anemia on HbA2 levels, and how should we interpret HbA2 results in cases where iron deficiency has not yet been corrected? Would you recommend delaying the diagnosis until iron status is restored, or are there alternative approaches to ensure accurate screening in such cases?
Yes, that is a very good question. I must admit that in the Netherlands, iron deficiency is less a profound problem than in many other countries, probably because of nutritional reasons, even though in pregnancy you also have, of course, iron deficiency, and that is not different with the pregnancies in the Netherlands. I think it's a very valid question. Usually what we do when we are requested for carrier detection is that we do a first screening for a gap PCR. So alpha thalassemia is the most usual reason for a microcytic hyperchromia. However, that is because the A2 is normal. But and if we see an iron deficiency, usually we stop there for the moment, and then we report back, please adapt iron medication, so that, and send in again if there is a suspicion, still a suspicion for thalassemia. The reason why we do it in this way is that the patient comes with a problem, and the problem is anemia. And you want to solve the anemia. And you cannot solve the anemia by saying you have thalassemia, because that doesn't change anything. However, addition of iron will help even a thalassemic patient to have a suboptimal condition. So from the clinical point of view, the advice is first do iron suppletion, and then you do, if there's still a microcytic hyperchromia, you extend the analysis with thalassemia research. Of course, if you have a request from the genetic point of view, someone wants to know if he is carrier because his partner is also carrier, then we proceed immediately to the DNA analysis, even if the A2 is borderline. So yeah, that is how we do it.
Okay, thank you very much, Dr. Harteveld. So actually, this is very interesting because we found these cases very common in Indonesia, and because you already mentioned that if we want to wait for iron supplementation, maybe we'll take longer time, and then while the patient needs immediately to have the diagnostic to get the exact treatment. So yes, we sometimes we also directly going to the genetic analysis, not necessary to wait for the iron supplementation completed in the patient. Begitu jawaban dari Dr. Harteveld ketika kita menemukan kasus suspek thalassemia dengan HbA2 menurun dengan kecurigaan ada defisiensi besi. Di sini memang secara algoritma memang kita harus mempertimbangkan untuk melakukan suplementasi besi dulu, dan mungkin ini sekitar 3-6 bulan. Apabila sudah terkoreksi, memang kemudian kita melanjutkan kembali ke HB typing seperti HPLC atau capillary electrophoresis. Tapi apabila keadaan pasien tidak mendukung untuk penundaan penegakan diagnosis lama sekitar 3-6 bulan, mungkin kita bisa langsung menuju ke analisis genetik untuk mendapatkan diagnosis yang lebih tepat dalam waktu yang lebih cepat, dan juga agar pasien bisa mendapatkan penanganan atau terapi yang lebih cepat juga. So actually we already reached at the end of our discussion session. On behalf of Department of Clinical Pathology and Laboratory Medicine and Thalassemia Research Group Universitas Gadjah Mada, I sincerely thanks to Associate Professor Dr. Cornelius Leonard Harteveld for his impressive sharing experience, and then please give applause to him. And also, I would like to thanks to our participants to keep enjoying this activity of this lecture. And then by having this lecture, we hope that we can give contribution to thalassemia in Indonesia, and then to make it further collaboration, and then also make more advancing diagnostic care in thalassemia in Indonesia. By this, I would like to officially transfer this session to our Master of Ceremony, Dr. Haris. Have a nice day, and thank you.